In today's tutorial, we will discuss about the classification of the chromatography. Before we move towards the classification, I would love to elaborate the term chromatography a little bit. After that, we will explain the classification, the different classes and the different types of the chromatography. So let's start from the introduction of the chromatography. Chromatography, what is chromatography? It is actually a separation technique that is used to separate the components of a mixture. Suppose we have a mixture and we want to know the components of this mixture, and we are just separating the components of this mixture. So the technique by means of which we separate the components of this particular mixture, that technique is actually known as chromatography. And we have different types of the chromatography. Now we know what types are actually present with us. So after that, the very next point is the main components of the chromatography. In every chromatography, we have two very important components. Those are the stationary phase, number one. Number two, the mobile phase. These two components are must and very important in each type of the chromatography. Now, what is stationary phase? Stationary phase stands for the standing phase, stable phase, stagnant phase, fixed phase, whatever the word you understand. In short, our stationary phase is stagnant, standing, fixed. One phase is fixed and another one is moving. The movable one is known as mobile phase. So we have one that is fixed, another one is moving. The fixed one is stationary phase and the one that is moving is known as mobile phase. So we have two phases. One is fixed, another one is moving. Stationary phase is fixed, the one that is moving is mobile phase. That's it. And the sample that we actually want to separate, this is actually separated by these two phases. How? Like, suppose we have a sample and in the sample we have components. Suppose we have 1, 2, 3, 4 and 5. We have 5 components in this particular sample. Now how these components are separated by means of this stationary and mobile phase? Very simple. Our component, when we just place it in between these two phases, what will happen? Some of the components, we have now 5 components. Some of the components will show high interaction with the stationary phase and some will show with the mobile phase. Those components which are showing interaction with the mobile phase, they will start moving with the mobile phase, and those which are showing the interaction with the stationary phase, they will also start moving but slowly and might stick to the stationary phase. So like this, what will happen? The components present will start separating by means of the stationary phase and the mobile phase. Those having high interaction will move with mobile phase. Those having high interaction with the stationary phase will just move slowly with the stationary phase. So that's it, the very simple logic behind how our sample is separated. Now let's come towards the classification. We have 1, 2, 3 different classes, and we do have further types in each class. In this chromatography class, we have number of other types of the chromatography. In the second class, we have further more types, and in third class, we have further more types of the chromatography. Let's come to the first one, based on the stationary phase and mobile phase. Now these all classes are actually based on our stationary phase, mobile phase and sample. How? Like, let's know. Stationary phase and mobile phase. Remember, we have a solid stationary phase and we have liquid stationary phase. And a very important point is that we don't have any type of the gaseous stationary phase. Our stationary phase must be solid or liquid. It cannot be gaseous in nature. And the next thing is that our mobile phase can be liquid, a gas, and it cannot be solid. Very important point, must note it. Okay. Now, due to these two phases, we do have further types of the chromatography. If our stationary phase is solid and mobile phase is liquid, we have a type of chromatography known as solid liquid chromatography. And the same is the case again. And if we have solid stationary phase and gaseous mobile phase, that type is known as solid gas chromatography. And if our stationary phase is liquid and mobile phase is liquid, then the type of the chromatography will be known as liquid chromatography. And if mobile phase is gaseous, then that type is known as liquid gas chromatography. And the next type is based on polarity. Our stationary phase may be polar and it may also be non-polar. And the same is the case with the mobile phase. It may be polar and it may be non-polar. So in most of the cases, we have polar stationary phase and non-polar mobile phase. Now, in most of the cases, we have this type of the chromatography, and that particular type in which we are using this arrangement, that is actually known as normal phase chromatography. And sometimes we do have the chromatography in which our stationary phase is non-polar and mobile phase is polar. In that particular type, we are actually doing the reverse of this normal phase. So due to this reversal mechanism, means before our stationary phase was polar, now it is non-polar. And before our mobile phase was non-polar, now it is polar. So we just reversed the arrangement. Due to this arrangement reversal, this type of the chromatography will be known as reverse chromatography. Reverse chromatography. I hope it makes sense. Next one is classification based on supporting medium. You can also say based on geometry, and you can also say based on chromatographic bed. Now this is actually telling us if we are doing the chromatography on particular surface, and we do have two types of surfaces on which we actually perform the chromatography. One phase is planar, another one is column. If we are using a column for a chromatography, then that type of the chromatography is known as column chromatography. If we are using column. And if we are using any plane surface for the chromatography, then that type of the chromatography is known as planar chromatography. And we do have another types in the same planar chromatography. Number one, if we are using paper as a plane surface for a chromatography, then that type of the chromatography is named paper chromatography. And you can call it as paper planar chromatography also, because paper is also a plane surface. And we do have another one, that is TLC, thin layer chromatography. What we are doing in this we are actually applying a thin layer of the stationary phase on any particular glass or plastic. So this type of the chromatography is also planar because we are using a plane surface, and we are applying our stationary phase on the plane surface. So in short, the chromatography that you are actually performing through the plane surface is known as planar chromatography, and if you are using column for a particular chromatography, then that type of the chromatography is known as column chromatography. And that's it about the second class. Now let's come towards the third class, that is the principle, based on principle. Now what is the principle? You know, your sample, the way your sample is interacting with the mobile phase and stationary phase, that is actually called as principle. Now we have adsorption, a principle, partition, size exclusion or gel permeation, and ion exchange. Now what is adsorption? Now this is actually the way how our sample is interacted with the stationary phase and mobile phase. If we take our stationary phase solid and mobile phase liquid, our stationary phase is solid and mobile phase is liquid, and we put sample in between them. Now remember one very important point. You have a stationary phase which is solid and your mobile phase is liquid, and your sample that you are putting in between is now going to be separated by this stationary phase and mobile phase. Okay, so what will happen? One very important rule: whenever a gas or a liquid is adhering to the solid surface and then it is moving or it's staying on the solid surface, this adhesion is called as adsorption actually. So what is happening now? Now our sample is actually adhering to the solid surface, which is solid stationary surface. So this type of adhesion is called as adsorption. Now our sample is actually adhering, or you can say adsorbing, to the solid phase, due to which that type of the chromatography is known as adsorption chromatography, and that is functional through the principle, that is adsorption. You got the concept of adsorption, that when any liquid or any gas is adhering to solid surface, that is called adsorption. The next one we have is partition. Now what is partition? Our sample is actually doing partition between these two liquid phases. Now both the phases are liquid, okay? So a sample will be actually partitioned between those two phases. So this is the way how your stationary phase and mobile phase is actually taking these samples into parts. So it is making the parts of your sample. So whenever we are placing our sample into liquid phases and then that is separated, that type of principle is called as partition, and the type of the chromatography is known as partition chromatography. The next one we have is the size exclusion or gel permeation. According to this, our stationary phase is solid and porous in nature, and our mobile phase is liquid. What is happening in this? In this, our sample, which may be of different sizes, okay? Suppose we have here 5 components. Now in these 5 components, number 1, number 2 may be of large size, 2, 3, 4, 3, 4 and 5. These may be of small size. What will happen? Now these components, when they are passing through this porous stationary phase, which is a solid phase, what will happen? The large 1, 2, which are large size particles of the sample, they will move out or elute out with your mobile phase. And if you're talking about the 3rd, 4th and 5th component, what will happen with them? They are of small size. So we have here porous stationary phase, which is actually specifically made porous for the small size particles. So now here we were having how many components? 5. So the small components out of these 5 will somehow stick in these pores of the stationary phase, and those large size components will pass beside the stationary phase. So we have this type of our stationary phase. We have a space available for the large size particles, and these beads, you can say somehow beads, that actually pose in them, and they will actually give a kind of space for small size particles, and they will not allow the large size particles to adhere to them, so that size particle will move. How, like suppose this is the small size particle, it is coming, so when it comes to the bead, it will stick here. And meanwhile we have a space in between two beads or mini beads. So what will happen, the large size particles, if they are coming, so they will not stick here, they will move beside these beads. So like this, our sample is actually separated based on the size. So whenever your sample is separated based on size, that type of separation is actually done by the principle known as size exclusion or gel permeation. And the type of the chromatography which is actually separating the components based on the size, that is known as size exclusion chromatography or gel permeation chromatography. And we do have explanatory mechanisms in which we are actually explaining this entire mechanism in different videos. You can search it out if you want to learn more. The next principle is ion exchange. Now what is the main mechanism behind? Here your ions are actually doing the exchange. So we have two types of the ions, the cations and the anion. The cation, you know, it is positively charged ion, and the anion is negatively charged ion. What is happening according to this principle? According to this principle, we are actually having two types of the exchange. One is known as cationic exchange, another one is known as anionic exchange. What is this principle talking about? Here, if we have a charged sample and we want to separate that charged sample into components, then we will actually separate our sample through this particular principle. Now what is the short mechanism behind how it is doing separation of the charged sample? So whenever you just take your charged sample and you are just loading the charged sample on your this particular stationary phase, you know this stationary phase is also charged. So what will happen? This stationary phase will interact with your charged sample, then it will separate your charged sample into components. So in short, your charged stationary phase is interacting with the charged sample, and then it is separated. So if you want to know more about this, we have explained this particular type in another video. So I hope you got, and if still you have any kind of confusion in these particular types, you can drop your question in the comment box. We'll come for the answers very soon, and thank you for watching.